right, so thank you everyone for joining today. For those of you who don't know me yet, I'm Alex Lemens, and the director of our Molecular and Genomics Informatics core. We also have Pat from the Genomics Center on the line, and then the team from the Curio Biosciences, who are here to talk about their technology for spatial transcriptomics, which is going to be a lot of fun. But before I turn it over to them, just the general housekeeping items. If you have any questions, please drop them into the chat in the Q&A box. We'll monitor it for anything that we can answer on the fly. Anything else we'll answer at the end. With that, I'll turn it over to Jane and Bertrand to talk about their spatial platform.
Great, thanks for the introduction. So just note that I'm Bertrand Young, Director of Business Development for Curio. I'm currently in a hotel, so I'm not sure how good the Wi-Fi is, so I'm going to turn off my video for the rest of the presentation. Yeah, so we have a platform called Seeker that we just launched a couple of weeks ago, and we'll be talking about that here. So just a little bit about our company. We're a life sciences tools provider that's located in Palo Alto, was founded in early 2021. And a lot of the founders and upper management have come from various backgrounds in industry, all very experienced and leaders in their own right. But they have a lot of background in single-cell genomics and molecular counting. And part of the mission for the company as a whole is to offer a very robust spatial genomics solution that's at high resolution, and to really lower the barrier of entry for those researchers who want to run this kind of assay. So as many of you know, there's been a huge explosion in spatial technologies across the sequencing space and FISH space over the last few years, and as they have started coming out, there's been some challenges that have become apparent, some of which are listed here. But there really seems to be a trade-off between resolution and number of targets depending on the platform that you are looking at. And a lot of them are a little bit difficult or very difficult to run because of the workflow, and therefore it is not that easy to implement for a lot of labs or cores, and therefore not very amenable to high throughput projects or atlasing projects. A lot of times also, because of the need for fixation, there's a lot of tissue-dependent assay optimization that's needed. And there also tends to be somewhat high assay-to-assay variability depending on the platform that you are running. Additionally, many of the platforms also require the purchase of additional capital equipment, and that can be very expensive for especially smaller labs or even cores that are trying to run various assays in the spatial space. So what we're trying to do as a company is address all of these issues and provide an assay or solution that is very easy to use and can be implemented by anybody in any lab with standard lab equipment. So our Curio Seeker product is based off the Slide-seq v2 technology out of the Broad Institute. And ever since we onboarded this assay, we have improved it quite a bit just to make it more robust. But a lot of the features that you may have read in the original Slide-seq papers carry over into our product. So here you can see that we still have a 10 micron resolution for the assay. All the beads are 10 micron beads. They're very closely packed in together so that essentially there's no space in between them. You may have noticed in the Slide-seq paper, if you've read it, that it requires the sequencing of the beads so that you can determine where all the bead barcode locations are. But what we have done is we have implemented this process into our manufacturing line so that we do all of that sequencing in-house, so that all the end user will get is a text file with a white list of all the bead barcode locations. And what you'll do, as I'll mention later, is that you'll put this list or text file into the primary analysis pipeline along with your FASTQ, and it'll do all the deconvolution and give you a matrix file output from the pipeline. The current product right now has a 3 by 3 millimeter tile area size. And the plexity of the assay is basically unbiased. It's whole transcriptome with a polyphy capture. Recommended sequencing depth if the tissue covers the whole tile is about 200 million reads. Obviously, if your tissue is more transcriptionally active, you'll probably have to sequence more, but based off the HTML report that you'll receive out of the pipeline, there is a metric in there that you can determine whether or not you need to sequence more. So if you do, you just sequence it again and concatenate all the data sets. As for specialized hardware, there isn't any. It's just your typical single cell hardware that you're probably used to. So tape station, PCR machine, sequencer. There isn't anything extra that you'll need to get on top of this specifically for our assay. The sample types that we support right now are fresh frozen. Throughput is really user defined. So I have a lot of experience in single cell from my time at BioLegend, running the Slide-seq assay. But the first time I ran this assay, I was able to do 6 samples at a time. We do have some users that are doing on the order of 10 or 20. Of course, they're much more experienced, but it shows that if you're very comfortable with it, you should be able to run multiple samples at a time. But the first time you do it, you probably will run just a couple and get used to the assay over time. The workflow is 8 hours with hands-on time of about 2 to 3 hours. And the software that we provide is a primary analysis pipeline that just converts to FASTQ along with your text file to matrix files, and also provides an HTML QC report. So on the HTML file, you'll get a set of QC metrics along with an unbiased UMAP and a representation of the reconstructed genes or those clusters from the UMAP on the tissue, and then also the 10 top differentially expressed genes that the pipeline has automatically identified. And the purpose of this really is just to see if your assay ran correctly, or if your experiment ran correctly. And then you'll obviously go on and do secondary analysis with other pipelines, not commercially, but other pipelines that are available to the public. So the matrix file output files that you'll get from the pipeline are just a generic matrix file for whatever secondary pipeline you'd like to use, but you also receive a Seurat file if you decide you want to use Seurat for your secondary analysis. Here you'll see the actual product. So on the left, there's a couple boxes. One goes in the -20, the other one goes at room temperature. And the pouch you see there is a pouch of all the tiles. That goes in the 4 degrees. And all of this is stable for a year from date of manufacture. And on the right, you'll see the product itself with the tile. So in the middle, you'll see the white patch, which contains all of the 10 micron beads. There's about 80,000 beads in that small 3 by 3 millimeter area, and they're mounted on this square piece of glass that itself is mounted on a blue piece of tape that isn't really that sticky. It's kind of posted little stickiness just to prevent the tile from moving around while you're mounting the tissue. But this piece of tape is itself mounted also in this white plastic cartridge that's about the size of a normal glass slide. But what you'll do with this cartridge or slide is put it into the cryostat ahead of time so that it gets to the same temperature as your tissue block. And hopefully this will play correctly over the internet. But what you'll do is you'll section your tissue in OCT. It's a 10 micron section. And what we usually do is after we section, we flip it over to prevent it from curling too much, because that will interfere with the melting process shortly. But because the slide is at the same temperature as the the tissue section, you're very easily able to move it over the tile, over whatever area you're interested in looking at in your tissue. And for reference, this is a mouse brain, which is larger than the tile, so you don't have to worry too much about tissues being larger than the tile. They can be as large as you want. You just need to know approximately where you want to place the tissue. But as you see, you melt the tissue once it's in position, and what you can do after this is leave it in the cryostat while you're sectioning other tissue, or you can put it in the -80 for up to 4 days and process them all at the same time. We've tested that for brain at least, and the data looks exactly the same whether or not it's freshly prepared or stored in the -80 for 4 days. What you'll do once you get the tissue on the tile is you'll use a pair of tweezers or forceps to gently peel that tile off the blue tape, and then you'll put it directly into an Eppendorf tube with hybridization buffer and leave it in there for, I think it's 15 or 30 minutes. And then once you incubate that, you'll transfer the tile into another tube with RT mix for the RT reaction. And after that, you will add a tissue digestion buffer, which is meant to completely digest the tissue so that you have access to the beads. Or depending on your tissue type, you may have to pipette up and down several times to mechanically dissociate it further, just to make sure it gets completely into solution. Otherwise, it'll be more difficult to get the beads off the tile, and then also may interfere with downstream processes. But once you have gotten the beads off the tile, you'll take that glass backing out of the tube and discard it, and then the rest of the protocol is in PCR tubes or Eppendorf tubes. So it's pretty straightforward. The data that you should expect is shown here. So here is a mouse hippocampus. Depending on their tissue type or where you are within the tissue, you should expect varying amounts of UMI capture. So in this particular example, obviously in brain, there are areas that don't have that many cells and cell bodies, and the areas that do. So in the very hot spots where there are cell bodies and high density of cells, you should expect around 6,000 or 7,000 UMIs per bead, at least in this data set, which corresponds with about 1,000 to 2,000 genes per bead. But overall, for the whole tile, for this example, we're getting about 18 to 20,000 genes in total. And what you can do after this is take it into secondary analysis to look at individual genes where you have really good signal to noise. And because of the high resolution, you can see very clearly the structures of the tissue. As I mentioned before, this is for fresh frozen tissue, and because of that, there really isn't any optimization for different tissues or species. So, this assay is exactly the same protocol for whatever tissue you run or species. So we've had people run this on human, various mouse tissues, chicken heart, planaria, zebrafish, what have you. It all works the same. So here you can see an example of mouse liver, where the capture efficiencies are very comparable to what you saw previously. And again, you can see a very clear structure within the tissue just based off of the gene expression. And again, for mouse kidney, capture efficiencies are very similar. And again, you can see very clear structures within the tissue. Something more relevant to disease, we've also run this on human melanoma. So this is just an example of two of the outputs that you'll actually get on an HTML file. On the left is just an unbiased UMAP that we generate to cluster the data. And on the right is the mapping of all those clusters back onto the tissue. And so what we saw just right off the bat here specifically for this tissue is the title is completely wrong, but you'll see different genes with very clear, distinct areas of expression. So on the left, you'll see PMEL, which is a gene that's negatively associated with immune cell infiltration and can be a negative prognosis marker for that particular patient or sample. And what you can see is actually all the immune cells identified by the IgHG 1, 2, 3, and 4 is that it indeed is negatively associated, negatively expressed compared to the P-male. So anywhere that there was no tumor, which is represented by the P-male, there is immune cells that are trying to get in and infiltrate the tumor. On the right, there's another example of a gene called TMSB4X, which is crucial for melanoma adhesion and invasion. And you can see that it is also in the space where the tumor is not, and where the tumor is adhering and trying to invade the normal tissue. Another thing that I didn't mention yet is the ability to integrate single cell data that you may have with the spatial data. And there's a couple pipelines that are out there that can do this. One is RCTD, that was from Fei Chen's lab, that was specifically optimized for Slide-seq, so this should work pretty well. There's another very popular pipeline called cell to location that people also use for integration of single cell data. So those are just two of the options that are available for people to use. If you see on our website, there are a couple of videos. There's a previous webinar that we gave with the Stowers. The Stowers did a direct comparison of our platform, Curio Seeker, against the original Slide-seq V2 via a collaboration that they had with Fei Chen's lab. And what you can see here is that the Curio Seeker assay and kit is a lot more robust. There's a lot more UMI capture and much more bead retention throughout the assay. And so you just get much better data out of our kits compared to the original assay. And that's it for now. So if you have any questions, let us know in the Q&A and we can answer them.
Awesome. Thank you for the great seminar so far. It looks like we have someone raising their hand. Seema should be able to talk now.
Okay. Yeah, no, my quick question. So it's only for fresh frozen, right? That's what you said, not fixed?
Yeah. So we're doing fresh frozen right now. We're a very small company. R&D team is not that large at this point, so we just don't have the bandwidth to develop that right now. But it is on the list. We do get that request quite a lot for FFPE or at least fixed frozen tissues. So if we have the bandwidth to do that, we'll consider doing that.
Thank you.
Anybody else with any other questions?
I have a question. So, we often have faculty who are willing to be the ones to try something out. How would we work with you if somebody wanted to be, the volunteer to be the first one to try this?
Sure. Jane, do you want to take that question? You're on mute.
Thank you. I'm talking away. We have just recently come out of stealth mode. So we were in early access until just a couple weeks ago. So right now the product is commercialized. And if anyone would like to try it, they can contact me for a quote. I'm happy to do that. The kit comes with 8 tiles. So they will be able to do replicates or do a complete experiment for that. There is also a 1-year shelf life for the reagents. So if they don't have enough samples to do all 8 tiles at this time, they can certainly keep those, and there's no degradation of the signal or anything like that over time. So we are selling the product now. It's a commercialized product, and happy to talk about their experimental design or how they could best utilize the tiles for their experiments.
And can you provide hands-on training if people need it?
We have, yes, we have two things that we're doing right now. So we have a, protocol that I can give to people, and we have a tech supports department that can help out. We're doing a weekly training, going over the protocol in detail with people who are just getting started. And so our tech support scientists will go through in line by line and just sort of make sure that people are aware of where the safe stopping points are in the protocol, where they have to move quickly, things like that. And that's been super helpful for people to feel confident in doing the protocol themselves. So at this point, our tech support group everyone is located on the West Coast, except for me. I'm in the Boston area. So we don't have a field application scientist on the East Coast yet. That will be coming fairly soon. But at this point, it's a very standard sort of library prep. And so most people are comfortable with that, with a little bit of training from our tech support group.
Yeah, and that training is online, not in person, not hands-on.
Zoom call every Thursday. So if you want to come and see it, send me a message and I'll be happy to send you the Zoom link.
Okay, great. Thank you.
Sure.
Any other last questions?
Bertrand, I did want to mention that we are in early access for a larger tile size. So if you're using human tissue, or if you have just a need to have a bigger tile area, we are in early access right now with a 10 by 10, and that should be coming out sometime this summer. So if that's something that you're interested in, please get in touch. Oh, here's a little.
Yeah. So this is an example. This is actually a 9 by 9. But this is something that we generated late last year, and because it was so successful, we decided to move ahead with an early access 10 by 10 tile. So this is just an example of data. For your information, this is actually very shallow sequencing. So as you saw in an earlier slide, for a 3 by 3, we recommended 200 million reads, but for something like a 10 by 10, you probably need to be closer to 2 billion reads. But unfortunately, we don't have a NovaSeq in-house, so we could only do a 200 million read run on this for this data set. But you can still very clearly see individual genes, even though it's very shallow sequencing. So we were very happy with this, and people are starting to test this out now.
All right. Cool. Well, then, thank you very much for the great seminar. If nobody else has any more questions, we can end it there for the day. We'll have the recording up and posted over the next couple of days. But if anybody has any questions, feel free to reach out to myself, Pat, or anybody on the Curio team, and we'd be happy to help. But with that, thank you very much, everyone, for joining, and enjoy the rest of your day.